I'm an agricultural science teacher. This is a tutorial offering support to Leaving Cert students in relation to the Individual Investigative Study, the IIS, particularly for the class of 2024, where the brief is, as outlined here, the role of food production in maintaining natural resources in Irish agriculture. I have uploaded similar content in the past, and I know a lot of students found this really useful. I've had a lot of requests to provide something more updated for this current year group, and I guess that's what this is aiming to deliver. From the off, can I just state that this here is really important. It is your ability to carry out scientific method that is being examined here in the IIS. The brief above is simply a theme that you must align with throughout your report, your investigation. But the more you demonstrate good scientific method, the more marks you will obtain. Put simply, this is less to do with food production or natural resources and a hell of a lot more to do with your demonstration of your ability to think and work like a scientist. So don't allow yourself to get too bogged down or distracted. Don't let this become a project about natural resources. It is not a project. It is a report on your ability to think and work like a scientist, whilst, yes, following this theme of food production and natural resources in Irish agriculture. You should all be familiar with the layout of the marking scheme or the report. This is just a suggested template. This is a suggested template I've compiled from experience and various other sources, and I know that this has worked well in the past, so we'll continue with it. You need to provide an overall title to your investigation, to your report, I should say. This is separate to your experiment title. This is separate to your research question. This is an overall title. I would suggest you try to keep it concise. Think of it as like what you would put on the cover page if this were to ever appear at a bookshelf in a library or wherever and someone was passing by. What is it that's going to grab their attention? What is it that's going to sum up the contents of this investigation or this report? So it could be something simple like maintaining Irish water quality in Irish agriculture, or it could be something simple like minimum tillage versus conventional tillage, which is more sustainable. Something along those lines. There's no wrong title. I suppose it's just about having an effective title. You do need to provide a topic. So this is where you'd state briefly what specific area you intend to focus on, just drilling down a little bit more than your title. So you might start explaining, what area you're interested in, why you're interested in it. I would advocate that you definitely choose something that you have easy access to or even better hands-on experience of. There's no point in choosing a topic that you really can't access or get much exposure to. Other than that, feel free at this point to start building in some references. There's research to be done as part of this introduction, and at this point you could start providing some references or research points that have helped you decide on your topic. You then have to provide a chosen agricultural enterprise. So identify clearly your chosen enterprise. This could be a spring calving dairy farm. This could be tillage farm. This could be any number of things, but to say the tillage industry, that's not a chosen enterprise. To say a dairy industry, that's not a chosen enterprise. Okay. Discuss your connection with the enterprise or your reasons for choosing this enterprise. I would suggest you provide a general location of the enterprise. Remember, there are 10 marks going for communication and innovation, and the whole fact that it is individual and innovative. The fact that you're basing it on a real farm and you're suggesting where it is and whatnot, the other enterprise on the farm, that all helps in that regard. A research question then. You need to clearly state your research question. In doing this, I'd ask you to consider the following. Good research questions tend to allow for different viewpoints to be researched, kind of like the pros and cons, the for or against. Your research question is something that you must be able to deliver on in the remaining part of the school year or growing season, in a suitable time scale. It's not something that's going to take way too much time that you don't simply have. A good research question enables you to conduct a controlled scientific experiment, allows for the collection of primary data, your own data, and of course allows you to follow scientific method throughout. Okay. There's no secret, the advent of ChatGPT and various AI bots like that. I have found it useful. I have lots of students who have good ideas and have tried to formulate their own research questions. And, working with the likes of those AI bots, I found it as a valuable resource in terms of formulating a good, clear, concise research question. You give it your idea, you give it your thinking, and it might just help you work towards a better, a more carefully worded research question. Moving swiftly along, the next important paragraph is connection to the brief. So my experience is that students generally often have a very good idea in their own heads what it is they're doing, and they have it all worked out, and that's great. But sometimes when I read the reports, I'm not personally sure, as an examiner or someone just reading it, what exactly is going on. So this is an opportunity for you to make it very clear to the examiner exactly how your investigation aligns with the brief. In this case, the role of food production in maintaining natural resources. So spell it out to the examiner that maybe it's a case of slurry is a byproduct on a dairy farm. Dairy is a significant component of food production in Irish agriculture, but that slurry is a hazardous waste potentially, and we must spread it on land at certain times of the year using certain methods, etc., in order to maintain our natural resource, which is water. You absolutely should now start feeding in some of your research points from expert opinion through your research. Use these research points to strengthen how your idea, your concept, your investigation aligns with this brief. Bottom line is, when the examiner reads this paragraph, they should be crystal clear on how your overall investigation is matching, meeting, or aligning with the brief. Similarly, you have to establish a connection to your chosen enterprise, or how your report, your investigation connects with your chosen enterprise. So I'd suggest here, again, these are all just suggestions, you identify maybe possible implications or the likely impact of your investigation that it may have on your chosen enterprise, that dairy farm that you based it on, or your tillage farm, etc. Might it mean you can farm more sustainably, reducing your carbon footprint? Could you bring any details or facts or stats or figures in to support that? Maybe it might facilitate a move to organic farming. Maybe you can reduce your input costs. Maybe you can maximize profits. Maybe it'll mean you'll produce more product. It would completely depend on your own individual topic, but you must show an awareness here of how this investigation might impact, might connect with, might change maybe farming practices on your specific chosen enterprise, the farm that you have based this on, or the enterprise you're basing this on. That's all worth 10 marks already, and I would suggest, and I'm going to make this point again and again, I would be a believer that every student can get 10 marks out of 10 there for that part of the introduction, if you follow that advice and you're diligent about it. In relation to research, there's another 10 marks going for the introduction section. Remember, the introduction is worth 20 marks. The research is worth 10 of those 20 marks. So my experience again is, I don't know, there seems to be a bit of a kind of a phobia about research by some students, not by others, but research can be anything that helps shape or guide your investigation. So I might choose to do a bit of research around types of food production in Ireland in line with the brief, or maybe different types of natural resources. I might look at statistics around the quality of relevant natural resources. What impact is slurry having on water quality, or tillage is having on soil erosion, or whatever the case may be. Maybe I've got some sort of a hunch as to what I'm going to do, but I need to be considerate about health and safety considerations that I may have to factor into my experiment. I need to research, what equipment do I need? When is the best time to do this? And so on. So research doesn't have to involve splitting atoms or landing on Mars or anything like that. Research can be anything that is shaped or will shape and guide you forward in this process. Research certainly is not a list of unrelated facts. Okay, there's absolutely no point in cutting and pasting research points and sticking them into your report. You can cut and paste, but where you do that, you must demonstrate your own understanding of your research points, or use those sections of reference points to strengthen what it is you're trying to say. Form your own opinion based on what the expert opinion of somebody else. You can, of course, conduct interviews. You can, with a suitably qualified person, be that a teacher or a farmer, online resources, science journals, reports, the list is endless. But please make sure at the the end of this introduction section, you have embedded somewhere in your introduction 7 to 10 minimum research points from a variety of different sources. So don't just, go onto Google and hit up Google or Chagask or whatever the case may be 10 times. Get a variety of different sources. From, of course, credible sources, reliable sources. Again, provide inline citations after each piece of information used. If you're not quite sure what that means, I might do an additional tutorial on inline citations or referencing, but certainly get help with that. You have to say, in the body of text where you're getting this information from, and then that ties in with your reference list at the end of the report. So that's it. That's your introduction done. And again, I know it's easy for me to say, but I'd be encouraging all students to be really hungry for those 20 marks. If you push yourself, I think you can all get the 20 marks in that introduction, okay? The research doesn't have to appear as a heading like this. You could, as I say, include research, build that into your connection to the brief or your connection to the chosen enterprise, or even in identifying your topic or so on and so forth. There is no harm also, by the way, if you want to put in a subheading showing some of the additional research that has guided you forward. Section 2 then is the investigative process. Up to 800 words and 25 marks. 25 marks out of 100, that's a quarter, 25% of the overall IIS. And I would be encouraging all students to chase down every last one of these marks. Okay, I think 25 marks are on offer, and I think most students should be looking to get the full 25 here. You need to provide experiment title. This is not the same title as you've given earlier to your overall investigation report. This is an actual experiment title. So state it clearly, and there's a certain, style or language to experiment titles. If you look at physics, chemistry, biology books or junior science books, you'll see, typically they'd start with an investigation to determine and whatever it is you're looking to investigate. One experiment is usually enough, but you might find yourself, I know I have some students who maybe are looking to investigate a couple of different independent variables, and I would always advocate each experiment should just have one independent variable. So if you find yourself wanting to investigate maybe two different things, how they may affect something, I would suggest you break that into two separate investigations, or part A and part B. To sum up, each investigation, in my mind, should only have one independent variable. More on variables in a little while. So once you've formulated a clear, concise, straightforward title, then have your list of apparatus. Leave nothing out, so whether you use a test tube or a tractor or everything in between, make sure you include that in the list here of apparatus, okay? Then you have a hypothesis and a prediction would be my suggestion. A hypothesis is a general suggested explanation to an observation. You may have learned or heard that it is an educated guess based on prior observation or experience. So I would always suggest keep your hypothesis general, okay? So it might be something along the lines of fertilizer application rate may affect crop yield, okay? Fertilizer application may affect crop yield. That's an observation, okay? Just to state also, your hypothesis should, of course, align with your research. You can't jump about. You've just done an introduction, you've formed a research question, you've outlined your title and so on. Now this investigation and its hypothesis should follow suit. It should be aligned with your research. Then you have your prediction, and a prediction, I suppose, is the actual outcome. The actual outcome that I would expect if my hypothesis above turns out to be correct. So I'm suggesting you state precisely what you think will happen. This is all future tense, okay? So for example, if the application rate is reduced by half, then the crop yield will also be reduced by about 50%. That's my prediction. Maybe I'm right, maybe I'm wrong, but it makes sense in terms of it is built off of the hypothesis above. It is connected, but it's precise. Then if you move on to health and safety, here you need to list all relevant safety risks and the associated necessary precautions you need to take to avoid those risks. It's a common mistake for students just to identify risks, okay, or maybe just identify precautions. You need to link the two. So I'm wearing safety glasses to protect eyes from dangerous chemicals, and follow that pattern all the way through. List all the relevant safety risks and precautions. This is a biggie, okay, variables. It's very important each student identifies clearly their variables. So you typically have control variables, independent variables, and dependent variables, okay? So control variables is everything that you are going to keep constant. That's not necessarily everything that stays the same. Okay, there is a slight difference there. Control variables are all the things that you fix and you ensure they do not change throughout the duration of the experiment. So maybe it means a constant temperature of 10 degrees, or maybe same sized plot of 1 meter squared, but be specific, state, what it is that you're keeping constant. Then you have an independent variable. As I said, the one thing that you will change, okay, during the experiment. So again, control variables are everything that you keep constant. You make sure they don't change. And then your independent variable is the one thing that you will change. Finally, then, you're going to measure something, and this is your dependent variable. Whatever it is you are measuring, whether it's the mass of the grass or the mass of the sediment in the water, or whatever the case may be, you're assessing something, you're measuring something, and that is your dependent variable. Your dependent variable. That's worth spending a bit of time on if you're not sure. Make sure those are well formed, laid out clearly, however you see fit. Then we have the method, and in relation to a method, again, I think this is something that you should spend a bit of time on and make sure you get it right. Outline precisely the steps to be taken. Okay, use numbered points, so step 1, 2, 3, 4, as opposed to just as opposed to bullet points. State the method in a non-personal manner, so it's not I did this or he did that or we did this. Okay, we don't use that language. It's just, again, best practice in terms of scientific method. So instead of saying I recorded the mass, you should try and reword that sentence to say, using a mass balance, record the mass. Okay, so it's non-personal. Use the metric system, so whether it's a second, litre, kilogram, grams, etc. Stick to the same, SI unit, the metric system. Include the when and the how. Was it daily, hourly, weekly, and so on. Make a big effort to use all the correct names of all the various apparatus or chemicals you used. You can, of course, include labeled diagrams or even photographs of the setup of the apparatus and so on. And a good test is if you hand your method to somebody and ask them to read it, and ask them to pretend they have to conduct the same experiment, if that person comes back to you with any questions, that means your method is not fully clear enough. So I should be able to hand you a set of steps, my method, and maybe you should be able to go away then and conduct the same experiment without saying a word to me and get the same results. That's what you're striving towards. Then you'll know your method is detailed enough, clear enough. But if somebody comes back to you and goes, well, hang on, do I do this or do I do that? Or which do I do first? Or how much of that? Or how do I set that up? Your method isn't detailed enough. Again, this is all about innovation. There's marks for being innovative. So sometimes less is more. Sometimes rather than doing something on a big field scale, sometimes I have students who are going off to do great things on a large scale, but very often they could have mimicked it maybe in the lab or in their garage or garden shed at home or in the back garden using a window box, etc. And sometimes those simpler methods are more innovative and you have a little bit more control over them. Okay, on to section 3, and this is the results, analysis and conclusion section. This is worth 35 marks, so worth the largest amount of marks overall, a maximum of 1,000 words for this section. So if we break this down to 3 subsections, so results first of all. The results put in its simplest form means what happened, okay? So state what happened during the experiment. This is most likely going to be just done in the form of presenting your data, using a data table or whatever. Use the appropriate method, so whether that's a data table, and then support that with a pie chart or a graph or a histogram or a line graph, whatever visual best represents your table of data, that's what you should use. Do not present the same data using different methods. Don't put it in a pie chart and in a line graph and in a bar chart. That's too much, that's overkill, and there's no extra marks. Choose the most appropriate method, because that's a scientific skill in itself. For example, if you're working with percentages, a pie chart might work best. If you're looking at maybe something over time, if time is a variable, you might use a line graph and so on. Include where possible. It's not always possible, but where possible, you might have qualitative data. Okay, so around the senses, was there an odor? Was there a smell? Maybe a taste? As again, it depends on what you're doing, but certainly everyone should be looking to have actual primary quantitative data, numerical data. That's the big one there. What else? In terms of data table tips, independent variable is typically shown in the left-hand column like you see in this sample table below, so you don't have to type in independent variable. In this case, this is just an example. Maybe my independent variable was application rate, and I will show the various treatments then under that independent variable: no fertilizer, half rate, full rate, etc. But again, I'm stipulating, I'm showing, I'm making it easier to read, to communicate, for the examiner to follow what's going on here. Application rate, I could have just went with plot A, B, and C, but I'm actually putting in here what plot A and C, what those treatments were. So that left-hand column there, that's all the independent variable. Then typically, a good data table will have across the rest of the columns the dependent variables. Okay, so in this case, in this working example, and that's all that is, grass yield, again showing the units, grams in this case, trial 1, 2, 3, and then the average here over on the right-hand column. Yeah, so data tables, I mean, this is quite a small, simple one. Some data tables can have, a high number of rows and columns, and, it's all there. But don't forget, that's why you choose a suitable visual. Like, you might spot a pattern or trend on a graph that you would never have seen looking at a data table. So, don't just take that for granted. Remember, your data table is like step one, and then you represent that with a visual on a histogram or a bar chart or a pie chart or a graph, so to speak. And it's those two representations that you will be using later in your analysis to spot the various trends. In terms of graphs, I always say to my students, it's time now for the best graph you've ever drawn. This is a Leave Insert subject. This is worth 25%. You're going to do a graph. If you're drawing it, if you're creating it on software, Microsoft Excel or otherwise, this is the one to pull out all the stops. So using the SALT acronym, S for scale, okay, so make sure your axes are drawn to an appropriate scale to show the data clearly. You do graphs in maths, you do it in science, various other subjects, I'm sure, no doubt. So make sure you get help from your teachers or from wherever, but this graph is the best graph you've ever produced. A for axes, both axes must have equal increments, so that's not shown on this sample graph here, but make sure that the increments between each measurement are equal on each respective axis. L for label, both axes with correct units, so make sure that whether it's kilograms or grams or seconds or whatever the case may be, that they are clearly defined on the graph. Again, that's not shown here, but it is mentioned here, so make sure you follow that. And then give your overall graph an overall title, I should say. Provide a clear title, and the title should explain the purpose of the graph. So it might say a graph showing the relationship between application rate and resulting grass yield. Okay, presented well, presented clearly. Use of color is always good, and make sure you have everything covered. Okay, there's no excuses on this. Okay, the best graph you've ever done, you're doing it now. Also, best practice to have the independent variable here on the y-axis, with the dependent variable shown on the, independent on the x-axis, apologies, and the dependent variable shown on the y-axis. Okay, so again, y-axis, dependent variable, independent variable on the x-axis. In terms of the next subsection here, analysis. Analysis is really identifying patterns, okay? So if you think of sports, many of you might be into sports, they might say to you, after the break we'll have the halftime analysis or the full-time analysis, where so-called experts sit around and analyse the match. What trends did they see? What way was one team playing or defending or attacking, or the case may be? So analysts spot patterns, spot trends. In your case, use your data, use your graph, your image to help you to do this. That's all we're looking for here in analysis. Okay? We all have strengths and weaknesses, but if you're striving towards, I suppose, excellent analysis, you need to be able to show good insight, good understanding, your ability to figure out relationships between variables. So whatever your experiment is, can you see any trends? Is a change to that independent variable causing a resulting change over here to this dependent variable? Simple things as a checklist, be sure to identify the highest reading on your graph, the lowest reading, the range between the two, calculate percentage differences where appropriate, calculate the mean, the mode, the median, standard deviation. Again, get help with from your maths teachers or whoever. It all depends on what level of analysis you're looking to provide. Identify any causation, any correlations that exist between the variables. That would be, in my mind anyway, excellent analysis, and it could serve there as a useful checklist to you. Try to explain some of these trends. This is where, okay, you've identified the trends and now you're trying to provide reasons for the why. Why have these trends established themselves? Could it be, could you bring in some of your knowledge from your course or from your experience or some of your research? Maybe there was an increase in yield because the extra nitrogen in this case, and this is just my example, led to a greater amount of chlorophyll being produced. More chlorophyll meant more photosynthesis meant greater yield. It'll all completely depend, of course, on what it is you're doing. But I hope you can see from this example, you're trying to explain the trend here. Again, you might even, and the whole purpose of this is, to get you thinking and working like a scientist. And it'd be fantastic if you, somewhere along your journey, realize things. You say to yourself, well, actually, I think that might be because of whatever the case may be. That's really good analysis and shows detailed knowledge that you've gained through experience and studies. Discuss maybe briefly if it aligns or not with other investigations done by other experts. Any calculations that are required, you'd put them here in this section also. The final subsection to this section is the conclusion. I know students often get confused between results analysis and conclusions. Again, just to recap, apologies if I'm repeating myself, the results is exactly what happened. No explanation. There's my data table. There's my graph. That clearly shows what happened. Your analysis, what were the trends? What were the relationships that are obvious from looking at this table of data and this graph? Now my conclusion is maybe where we start to use that analysis, start addressing our research question, bringing in any other evidence from outside of that, maybe from the ag science sector, using secondary data to strengthen your conclusion. So my data showed this, however, this contradicts other research done by whoever, some expert, or perhaps my data is actually supported by this secondary data I obtained from wherever. At this point in your conclusion, you need to decide whether or not you now agree with, can you confirm your hypothesis? Do you accept your hypothesis and prediction? Or maybe perhaps it's time to reject your hypothesis. Based on your findings, you now realize your hypothesis or prediction or both were incorrect. There is absolutely nothing wrong with suggesting now in your conclusion that you no longer accept your hypothesis. You've disproved your hypothesis. This is every bit just as powerful. It often leads to powerful discussion, and this argumentative sort of writing or discussion is to be encouraged. In your conclusion, I would suggest you discuss any limitations. In other words, anything that restricted your investigation. Whether that's sample size insufficient or whether there was issues around soil pH or nutrient level. Again, this completely depends on what it is you're doing. Maybe it's faulty equipment, weather, human error, any of that kind of stuff would all be considered limitations, and you should discuss limitations within your conclusion. Okay, so on to section 4, which is the reflection. Now, we've just in the previous section discussed the conclusion. Now, in life, I think when someone hears the word conclusion, so if I was making a speech and I finished up by saying, well, to conclude, I think everyone in the audience would assume, okay, that's it, it's over now, okay? So I get it. A lot of students, when they do their results analysis and conclusion, they pretty much mentally think that's it. And now we're coming along here and we're looking for a reflection. So I'd say to you, stop. Take a break, go away from it if you have the time, come back to it a week or two later. You should do your reflection fresh, okay? It is worth 10 marks, okay? That's worth the same, by the way, I would point out, 10 marks is the same as actually your results, your data table and graph. So you should put the same, I suppose, energy and show the same interest in a reflection. If you're doing it straight after conclusion, in my mind, it's never as good. So don't be tired with this section. Take a break and come back to it with renewed energy. It is worth valuable marks. So what goes into your reflection? Here you discuss maybe how well you feel your research question was addressed overall. Okay, it's a little bit more informal. It's a little bit more of a kind of a personal reflection on the process, on what you've learned and how you've developed and how well you think you did. Okay, so it's a little bit less formal. You might state whether you feel you addressed your research question well, a lot, a little, maybe why so. You might state if you're satisfied with your conclusion, what worked well, but more importantly, why it worked well. What went well in your investigative process? And why did it go well? What didn't go well? And if something didn't go well, don't be shy about it. Declare it. But more importantly, what has that taught you? What have you learned? Why didn't it go well? Reliability. Is your experiment repeatable by somebody else? Or was there insufficient research or an unreliable method that couldn't be repeated? Or were there inaccuracies maybe when taking measurements, etc.? You might choose to bring in here, like this list isn't exhaustive, these are just ideas, suggestions. You might acknowledge sources of errors. All kinds of things could be coming in there in terms of sources of errors: drought, illness, power cut, extreme weather. The list is endless. I would definitely encourage you to mention some sort of a modification in your reflection. So what would you now consider, what would you modify, what would you change about your investigation? And again, why and how would that make your investigation better? So what would you do differently to make it better? Then there is an extension here. I would definitely encourage you to say or include or discuss possible extensions. So an extension is more like, okay, right, so that went really well. Now, if I was to take this to another level, here's what I would do. If I was to extend this experiment, or based on what I've learned here, I think the next obvious step would be to go ahead and investigate, different fertilizers or different crops, or there should be a natural progression. Whatever your topic was, let's assume it's gone well and you've learned loads. Now, if you were to take this on to a new level in the future, where's the next obvious step? That would be an extension. Whether you're talking about modifications or extensions, and I would definitely talk about both, it must be evident that this will enhance and this should link with your original research question. You don't just jump ship now and go off and discuss extensions that have nothing to do with the overall brief or your own research question. There should be a natural flow to where you could take this or what you would change, okay? Anything else? No, I think that's enough in terms of reflections, just to emphasize 200 words. It's not a lot, okay? You need to be clear and concise and have a bit of impact there. That's worth 10 marks, not to be sneezed at. In terms of references, I've listed this as section 5. You must reference all external sources of information. You must give credit to those people who have provided information or research points throughout your report. There are various systems. I'm not sure how much you know about them. The Harvard system is one I personally use. There are many others. None of them are incorrect, but whichever one you choose, you must be consistent with it throughout. You can't chop and change the system of referencing. Okay? This is important. My experience is that a lot of secondary school students haven't got good experience of inline citations and referencing. As I might have mentioned previously, I will put together a little help video in relation to referencing and inline citations, so maybe check that out later. But just to cover this, you do need to have a reference list in your report. All references should be listed in alphabetical order. Don't number references, don't use bullet points, you just do it alphabetically. And of course, any citations provided throughout the body of the report must match with the reference list at the end. As I say, if you're not great on referencing, I will post an associated video on referencing. You could check that out, or otherwise ask around. Maybe you have an older brother or sister or parent who is quite familiar with various systems of referencing. Don't ignore it. It's got to be done. Whilst there's no marks for referencing, you'll note there's no marks allotted. If you don't reference correctly or citate correctly, marks will be deducted from the relevant section. So if in my introduction I haven't referenced correctly, well then it'll be marks taken off me in my introduction. Bottom line is, you got to do your referencing. Finally, I've listed this as section 6, communication innovation. This is worth the final 10 marks. Again, not to be sneezed at. So definitely, I think we're all guilty of making mistakes. Nobody's perfect. So you need somebody reliable to proofread your entire report. Okay, somebody who has a bit of an eye for this, who can spot spelling mistakes or grammatical mistakes or typos and so on. You and I, when we read our own work, we will think it's fairly good. We'll find mistakes, we'll fix them, and then we'll reach a point where we think it's right. It is totally worth it for you to give your report to somebody reliable to proofread it and ask them to find errors in it, okay, and fix them, of course. All your reports should be presented in a logical sequence. I've provided this suggested template. If you follow that, I think you can't help but have it in a logical sequence. That'd be my own opinion. The inclusion of data tables, graphs, relevant photographs, they will all help in terms of communicating and being innovative and improving readability. Definitely try throughout the report as you go through the various drafts, try to improve your use of terms, scientific language, agricultural language, have the correct names for things, try and use some of the appropriate terminology. This is a science report at the end of the day. Try to incorporate argumentative writing, like be critical. Critical means like you're questioning what maybe some of the research points show up, or your own data tables and graphs and what the trends are, and through your conclusions or reflections, you can be critical about many things, and that's to be encouraged. Nobody can teach you to be innovative. That's something that you're going to have to develop yourself over time. Some parameters, some basics to bear in mind. There is a maximum word count on your overall report. It is not to exceed 2,500 words. So that includes everything except your reference list at the end. So your reference list can be as long as you like. It is not included in the 2,500 word limit, but it does include titles of your diagrams, your images, your photos, etc. Just on that as well, there's guidelines up here, for example, 800 words here is the maximum for your investigative process. You can go over that slightly, and I would tell you to try and stick to the 800 words. What I'm saying down here is that you can go over the word count in any one section, provided that the overall word count is not over 2,500 words, okay? In terms of image count, it's only 10. That's not a big number, so definitely include photographs, but any diagrams, charts, tables, graphs, etc., they're all included also as images. They're included in that, so it is a bit restrictive. Some students in the past have tried to do collages where maybe they kind of collage together 6 or 7 photographs into one image, but that's been ruled out. That's not allowed, and every image must be labeled. So for example, image 1 showing plots A, B, and C. Image 2 showing fertilizer application method, etc. Whatever it is yourself. And also make it as easy to follow. You might have a photograph, you might have a label beneath it, an arrow pointing in at something, soil auger, or whatever the case may be. You might have, font size has to be Arial, font size 12. The maximum page number in your report is to be no more than 20. And for the class of 2024, the deadline is April 12th. Just then in conclusion, I suppose final words. In order to score full marks in each section, you must deal with all those subheadings within their correct section, as I've outlined in this tutorial. What I mean by that is you can't, put in analysis into the conclusion and expect to get marks for your analysis there. All your analysis goes in the analysis section, and that's where the marks for the analysis will be awarded. You can't say something in your reflection that you were supposed to say in your conclusion and think that you'll get the marks for it there. In other words, everything has a home, and make sure that what you're saying is in the right section, as I've outlined above. I hope that's clear. Just to recap, on the marks, can I just flick back up here quickly? The introduction there is worth 20 marks. That's a fifth. I personally think if you sit down, you follow this guideline, you go through these headings, you work through it, you take the advice on board from yourself, from your teacher, from whatever you can get here, I think, you should be aiming for the full 20 marks there. Make sure you have your 7 to 10 pieces of research from a variety of sources that's relevant, that you've explained. I see no reason why you won't get 20 marks there. Going on to section 2, the experiment itself, 25 marks. Again, can you come up with a clear title? List your apparatus, form a hypothesis and prediction, list health and safety. Sit down, carefully identify all your variables, have somebody check them, make sure that they are correct, and then submit them. A detailed method that somebody else can follow. A clear labeled diagram or photos of the apparatus setup. If you've done that, again, I don't say this lightly, I'd be advocating, I don't see why you don't get the full 25 marks there or thereabouts. Now you're up to 45 marks. You're almost halfway through the IIS in terms of marks already. 45. Then we move on to the results section. Results section, analysis conclusion is worth 35, but the results I know is roughly about 10 marks of this 35. So if you can present a simply well-drawn data table with a suitable image such as a graph, a representation such as a graph, following this advice here, you're going to get 10 of those 35 marks. Now you're up to 55 marks out of 100. I firmly believe most students can gain the 55 marks so far. When we get to the analysis section and the conclusion, okay, this is worth 25 of the 35 marks. This is where, okay, some students have strengths and weaknesses, we all do, but some students may find this a little bit more challenging. Some people may work harder at it. Maybe some marks will be won or lost here, but out of 25 marks, if you follow these guidelines, I'd advocate out of the 25 going here, I think you should be able to get 10, 15, 20, that's up to yourself. Okay, so we had 55, throw on another 10 or 15, you're up to 70 out of the 100 marks, you've got them already. Your reflection is 10 marks, again, easy for me to say, but come back to this fresh, follow it step by step, work through it. Take this advice, 10 marks again, now you're up to maybe 80 out of the 100. In terms of communication and innovation, you can only try your best, but if you follow this suggested template in a logical manner, using correct imagery or graphs or representations, etc., again, you'll get some of those 10 marks. So when you break it down, I would certainly try to do my best here as I conclude to encourage you all that it is attainable. Okay, don't look at it as a whole. Take it section by section. The marks will add up, and if you stick to this, remember, this is all about scientific method. How good are you at working and thinking like a scientist? I started with that and I'll finish with it again. This is very little to do with natural resources or food production in Irish agriculture. Yes, that's the theme, the discussion point, the lens through which you're working. But at the end of the day, each candidate is being assessed on how well their report shows them as working like a scientist, following scientific method. Don't ever lose track of that. That's it from me. I hope you found this useful. My email there is reviseagscience@gmail.com, or you can follow or subscribe on Facebook or YouTube. Anything I share will be through those channels. But should you need to contact me, you can get me on that email. I do update things from time to time, so yeah, keep an eye out. Okay, thanks for watching and best of luck. Hope it was a good help to you.